Hello everybody, welcome to the webinar for the SBV Improver, a Systems Biology Verification Tool. This is hosted in association with the openphacts project. So today's webinar will be presented by Natalia Bukharov from Solventa. So she'll go along and explain how the SBV Improver works and what you can do with that. So there'll be a chance for questions at the end of the webinar. You can type those into the chat module at the bottom of the GoToMeeting software, and we'll answer those at the end of the presentation. Okay, over to you, Natalia.
Thank you for the presentation. Hello, everybody. Good morning and good afternoon, wherever you are. I hope you have a better weather than we do. Here in Boston, we have almost a foot of snow, and it just keeps coming down. So I hope that my internet doesn't quit on me. So today I'm going to talk about Improver. It's a series of challenges, and they kind of similar to challenges you might have heard, Dream challenges by IBM. In fact, Improver is being organized and run by IBM and PMI and funded by PMI. Okay. Oh, somehow. Okay. The outline of this presentation. I will talk about Improver, why do we need Improver, why do we need crowdsourcing. Then I will talk about different challenges within the Improver, the diagnostic challenge, species translation challenge, and network verification challenge now underway, and that all is going into grand challenge. What is CBV Improver? CBV Improver stands for Systems Biology Verification combined with industrial methodology for process verification in research, which is kind of a complicated concept, but I will explain it a little bit more later, and it's really not that complicated. It's just a complicated name. So why do we need Improver? We need Improver and we need challenges like Improver because there is a lot of data out there and people analyze data using their own methods. What's interesting that every lab method seems to be better than an average. A couple of years ago in molecular systems biology, there was a publication that analyzed publications of methods for transcriptional profiling data analysis, and they found that 87% of researchers claim that their method is better than average, which doesn't seem to add up. And why that happens? It happens because people tend to select only specific studies in a niche that they are looking at. They tend to, they have usually training and the test sets at the same time. They tend to cross-validate and they develop this perfect signatures and don't work anywhere else, but they work perfectly on the data set that they are selecting. So the goal of the Improver is to divide a research pipeline into building blocks. A typical research pipeline in industry, at pharmaceutical industry, would look like you collect samples, you do the profiling, you discover patterns, and then you get an insight. You get a signature of a drug response or drug resistance, or a signature for a disease and for healthy. The Improver divides this into research block and tries to have a challenge for each of these blocks and then combine these challenges at the end for a grand challenge that will be identification of biomarkers. Each block is verifiable by a challenge, and building blocks support each other toward the final goal. And all this done is through the crowdsourcing. What is the crowdsourcing advantages? We can get many contributors with independent methods. We can get different solutions for various aspects of complex problems. Combination of different solutions are often better than each single method alone. And we can have unbiased benchmarking. This approach also complements the classical peer review process. The crowdsourcing used to be an academic pursuit at some point, but recently it became also a tool used by industrial companies, by pharmaceutical and biotech companies. One of the examples of the crowdsourcing challenge is the Behringer challenge that they conducted recently. They have published a huge number of molecules associated with different structural characteristics ranging from molecular size and shape to chemical composition, and they challenged scientific community to predict the mutagenicity of a new compound. They had 796 scientists who enters, and each scientist entered more than one solution to this problem, and at the end got an algorithm that was 26% more accurate in predicting the mutagenicity than the algorithm that was used by the company before. Another example of crowdsourcing, it's a collaboration between pharma and academia. Recently, several pharmaceutical companies selected 58 compounds that were proven safe, so they went as far as phase 2 of clinical trials, but these compounds then were somehow deselected as they were not effective for the indication that they were tested. So these compounds were then given to NIH, and NIH has devised this challenge where academic scientists could apply for a grant to conduct a research, preclinical or even a clinical trial to test these compounds for different indications. Right now there are 9 NIH-funded projects for this safe compound that could probably result in some new drugs, some new choices for some rare disease. Back to the improver. So as I mentioned, the improver has several different challenges. The first challenge was a diagnostic signature challenge, then was a species translation challenge. Right now we have a network verification challenge ongoing, and all these blocks will be later used for the grant COPD challenge. Diagnostic signature challenge. This challenge has already been completed. So this challenge aimed to verify different approaches, computational approaches used to classify clinical samples. Samples for 4 different diseases: psoriasis, multiple sclerosis, chronic obstructive pulmonary disease, and lung cancer were deposited for scientists to analyze. It's transcriptional profiling for these samples, and these were training data sets. Training data sets were public data set that were available from GEO, and they were well annotated, so they were used for this challenge. In addition to this training data set, PMI also offered independent test sets to test these signatures to predict which sample came from someone with the disease and which sample came from a control. There were 55 teams that participated. Submissions were scored by IBM Computation Biology Center and then independently reviewed by Improve Scoring Review panel. The take-home method was that combination of different approaches performed better than each individual method. And what's interesting, that method that worked well for one disease didn't necessarily perform well for another disease, and it seemed like an individual methods would be required for each particular disease to get the best results. Another take-home message that I thought was important from this challenge was that the selection of features, the methods, how the features were selected for analysis, made bigger impact on the accuracy of prediction than the methods that were selected for sample analysis. As I mentioned, there were 54 teams that participated, different teams from around the world, and the result of this challenge was published, and the winning team, and they got a prize. The next challenge was a species translation challenge. This was a more challenging challenge from my point of view, and the aim of this challenge was to understand whether we can translate results from rat to human. So the challenge had data from rat and from human cellular model, in vivo model, cell different perturbation, transcriptional profiling, and phosphorylation data. There were several sub-challenges. In one sub-challenge, the participants were asked to predict phosphorylation data within one species, within rat. If you have transcriptomic and phosphorylation data for perturbation A, and then you only have transcriptomic data for perturbation B, can you predict phosphorylation? In another challenge, there was the prediction had to be made between different phosphorylation in different species. We had results for perturbation A posted, and for perturbation B posted, and we had to predict what's going to happen in human for perturbation B for phosphorylation data if you know the results for perturbation A. There was also the third sub-challenge where participants were asked to predict gene subsets based on transcriptomics and phosphorylation data, and then there was also specific networks that were built based on using the inference from the transcriptomics and phosphorylation data. So interspecies prediction worked the best. You can imagine that predicting from rat to rat is the easiest. Several groups found statistically significant correlation, and the best performing team was from University of California at Santa Barbara. Interspecies, prediction between interspecies was more challenging. Several groups did find statistically significant correlation. Phosphate perturbation within rat was found to be easier to predict at 5 and 25 minutes. That's probably because the phosphorylation data are more accurate at those time points. And species-specific network inference from rat to human, there was really not much correlation there. 5 teams found few correlations, but it was found to be very difficult to predict from species to species. And now we have ongoing network verification challenge. So networks were built. I work for Solventa, and PMI has hired Solventa as a consulting company to build these networks. The purpose of this network verification challenge is to engage the scientific community to review, challenge, and make corrections to these networks. And then these networks will be used in COPD Grand Challenge. What kind of networks? These networks represent important biological processes implicated in lung physiology and specific processes related to COPD. Since PMI is a sponsor of this challenge, they naturally are interested in COPD and what leads to COPD. There are several types of models that are included in this challenge. There are cell-specific signaling like macrophage signaling, lung-relevant macrophage signaling. There are physiological signaling models, for example, like oxidative stress, and there are some models that reflect canonical signaling, for example, like MAPK network. How these models were built, how these networks were built. To build each network, we have identified relevant papers that contain information, experimental results that can be extracted from them to code relationship between entities in a specific network. In addition, we also used data sets. These data sets are publicly available, and they represent physiological and pathological relevant to COPD, perturbation of tissues relevant to lung physiology. We had a data set, for example, transcriptional profiling data set of whole lungs treated with LPS. We had T cells, dendritic cells, macrophages, NK cells treated with cytokines like IL-4, interferon gamma, LPS, IL-15, had lung neutrophil data sets. So all this transcriptional profiling data were analyzed and then put through a reverse causal reasoning algorithm to identify the upstream controllers, the proteins, receptors that can control those particular genes. The reverse causal reasoning, it's an algorithm that is Solventa proprietary, and it's based on a huge database that was collected over a period of 10 years with information about perturbation of different proteins, receptors, agonists, antagonists, that results in a particular transcriptional signature, or that just results in a particular gene going up or down, like RT-PCR data. So based on that knowledge base, reverse causal reasoning of a transcriptional profile and data set can predict upstream controllers that can change the gene signature that way. Okay, how this network was built. So the literature data, the data from the literature were extracted and coded in biological expression language. The biological expression language is also publicly available and can be downloaded, and people from academia can also use it to create their own knowledge base on their own computer. And the advantage of this language is that it's not difficult to figure it out. It's human readable and it's computer readable. Since in this particular challenge we have biologists, mostly biologists who participate, being able to read a Bell statement is very important for them to make contributions to this project. I have here as an example a statement from a paper, transcriptional activity of TP53 protein increases level of caspase 8. And on the bottom of the slide you see how it's coded in Bell. It's transcriptional, it's transcript stands for transcriptional activity, P stands for protein abundance, H, G, and C, it's a namespace, Hugo gene nomenclature, TP53 is a gene name, and then increases protein caspase 8. So it's pretty straightforward. And on the next slide I have a couple of more examples of how the Bell captures a scientific finding in a computable and human readable language. Okay, so these networks models, they can be used for drug discovery, biomarker, and toxicity applications. Network verification challenge in a nutshell. So as I mentioned, the networks were built. They were built by Solventa scientists in collaboration with PMI scientists based on the literature identified by Solventa and PMI scientists. And using the transcriptional profiling data from appropriate data sets and using the huge knowledge base that Solventa has. Right now, this challenge, these networks are going through the verification process. And after the verification process is completed, which will happen at the end of February, there will be a group of scientists from Solventa and PMI that will summarize all the additions and comments that we got from scientific community. And then some of this questionable edges, comments that people do not agree upon, they will be discussed at the Jamboree session in March, where scientists, the top contributors will be invited to this Jamboree session. And there will also be scientists from Solventa, PMI, and invited experts in the field. At the Jamboree session, we will finalize all of the networks, and then they will be published and they will be deposited in a publicly available as a publicly available resource for other scientists to use. The grand challenge. The grand challenge, as I mentioned, will be using all of these pieces, all of the other challenges that I have presented before. So we'll be using our knowledge from the diagnostic signature challenge, from species translation challenge, and we will be using this verified networks to find COPD onset biomarkers. So as I mentioned, we will have all these previous puzzle pieces, newly collected clinical data, newly collected rodent data, and we will try to identify biomarkers and develop a comprehensive model of COPD onset. The data for this grand challenge are being collected right now. There is an observational clinical trial underway where samples are collected from control subjects, from former smokers, never smokers, and from people with COPD. There are also studies underway in emphysema mouse models, and a number of different samples are also being collected: bowel, whole blood cells, lung histopathology, transcriptomics data, lipidomics, and some of the other data. This grand challenge is scheduled to launch sometime at the end of Q2 2014, and the organizers are hoping to leverage the wisdom of the crowd to develop methodologies for predicting the prognostic impact of different stimuli on COPD. The ongoing network verification challenge models will be also used in this challenge. And hopefully at the end of it all, we will understand the biology of COPD better than we understand it now. Okay, I have that's the end of my presentation, but I also would like to show the actual website, if that's okay. Yes, okay, I'm sharing my screen here. There is an echo. Okay, so this is the Improver Network Verification Challenge screen. So this, the home page has the information and videos about this challenge, and here on the bottom you can see our crowd. This is the map of the world, and you can see how many people from different countries have joined this effort. We have 52 scientists from the United States, 1 from Canada. We have 9 scientists from Russia, and we have 5 scientists from India that are contributed. And there is 21 scientists from Switzerland and 6 from Germany. So it's a substantial crowd, and people are reviewing this and adding the comments, voting up and down evidences. If I look at the community, we can see what's happening as we speak. So few hours ago, we can see that somebody was reviewing neutrophil signaling and oxidative stress models. Somebody created a new edge in the oxidative stress model and created a new evidence in these models to expand the model. And there is neutrophil signaling, oxidative stress. Probably 2 people have been furiously reviewing 2 models there. I'm actually working with. And there is an Fe2L2 signaling model. The leaderboard here shows where people stand, how many points they get. It's a competition, so people get points for voting up and down and evidence. People get points for adding additional evidence to the edge. You can look at the networks. So these are all 50 networks that are included in this challenge, and they are divided by types. There is a cell fate, cell proliferation, cell stress, inflammation, tissue repair, and angiogenesis. All of these networks reflect some part of the biology of the lung, normal physiological responses to different perturbation, and also COPD pathological responses. And if you open one of the models like this, it will open in a window, and we can examine edges in this model. We have some of these edges with a person running. It means that this edge is somebody already voted on this edge. The edge that has a mark like this, it means that it had been voted by 4 people, and the vote has been accepted. The edge has been finalized. If we click on an edge, we will see evidence that was that supports this edge. This evidence is a statement from a publication describing the relationship that are coded in this edge, and we can go to the reference to verify it, and we can vote accept or reject. And that's what participants in this competition do. Some edges have a lot of different evidence that were pulled from the knowledge base, from Solventa knowledge base. You can see here that we can approve or reject this evidence based on this reference that is included. So back home, so we have the challenge rules, we have all of the networks, we can see what's going on with the community, and participants can also see what other people are contributing, and they can vote on what other people have added to this. And you can also see the leaderboard, so each participant can see where they stand in terms of this competition. Okay, there is a link also to the Mother Improver website. This website has additional information about this and all of the other signatures that people can look, and there are also a lot of supporting publications and training and videos that are available to the participants. Okay, I think I'm done with my presentation, and now I can take questions.
Thank you very much, Natalia,
Yes.
for that interesting presentation. If there are any questions.
First of all, I had a question about the teams. Does this last challenge have teams as well? Because you had said the previous challenges had teams of people, and I would think that is more difficult to get a team organized. And one person obviously doesn't want to compete, let's say, against a team.
Yeah, we have a couple of teams that are organized because they come from the same institution, but we have not really judged people by teams. We are mostly looking person by person performance, because in this case people are mostly contributing to what they know. There are people who are experts in macrophage signaling. They contributed to macrophage signaling, and it's hard to find 10 people that would know macrophage signaling to organize a team around a model.
Yeah. Okay, so when you said we had teams that compete, it was basically just, it was individuals that were competing and not really.
Yeah.
Okay.
There are individuals that are competing. Some people did organize themselves in teams, and if we had more teams, we would probably do something with that. But since we had very few teams, and in general we didn't really have that many participants, judging people by teams, I show you here on the screen. Here we got these few teams.
Yeah.
And these are the points per team. But these teams, somehow they comprise mostly of people that have not contributed that much. So we're judging people by individual performance. Because you see that these people contributed as 1,141 points already. It's a lot more than any of the team.
I see.
A few people contributed.
Okay. So I don't know, do you have a sandbox? Could you do a demo of how you do one of these annotations? So that we could just see how it works? I know you showed us the Bell model where one thing increases something else, but if you just had I don't know if your system has a sandbox, so things that are maybe just made up could be tried, and just to get a feel of how difficult it is technically to do the annotation.
Well, it's not difficult. We don't really have a sandbox. Usually when I do a demo, I have some examples. But right now I don't have an example handy. Oh, I picked up a bad bad edge. So I have one edge here that nobody voted on, and this edge here indicates that catalytical activity of CD40 increases transcriptional activity of the complex AP1. Okay. So what we would do is we would look at the paper, look at the quotation, then we would look at the paper. We will make sure that this acceptable species, and we agreed to include information from mouse, rat, and human. We would look to make sure that it's really a B cell, and then we would approve. And it gives us a drop-down box here, if you can see it, and you pick up why you're approving it, and then you write a comment, and then you just submit. And your vote is being recorded. Someone else can reject it because they find it that this was, some cancer B cell line maybe, and we shouldn't be including those in normal lines. But that's how the voting process goes. In addition, we can add new evidence. This particular edge only has one evidence. It would be nice to have additional evidence. So then you click on the new evidence, and it gives you a drop-down. It gives you a window on the left where it's also self-explanatory. You would add PubMed ID for the paper. You would record species where this experiment was done, and then it's either tissue or cell line, whatever was used. And then you include a quotation that will support this particular edge that you are trying to vote on. There is also a way of adding additional edges. So for that, you need to select an existing node in the model. Here I selected the TLR9 node in the model. We can add additional edge to this node by selecting on the right new edge, and then it will give you this window here where you have to select whether this TLR9 will be your subject or object. Let's say it's subject, and then you select whether it increases or decreases, and then you need to type an object. And when you start typing, then on the bottom you can see function, namespace, and entity. It kind of gives you a little bit of a cheat sheet what to use for Bell. If you are still not sure, on the right here you see there is how to make a Bell statement, a basic Bell term. It shows you how you are supposed to formulate it. It gives you some of the Bell terms here. And then if everything fails and you are still not sure how to extract a Bell term from the paper that you have selected, you can go to the convert text to Bell, and there is a window here where you can just paste the quote that you want to convert to Bell and submit it. And there is a magic fairy on the other end who's going to take that quote and email back a proper Bell edge.
Ah, okay. Okay. And so then you take that proper Bell edge and you put it into this form.
Yes.
Okay.
So yeah, people did make mistakes, but then that's why we are going to clean it up after this process is done, after the
okay, so the voting is only on the scientific relevance of it, not if you made a mistake in the construction.
No,
the Bell construction.
No. We are only judging people, ranking them based on the scientific merit of the papers that they contributed.
I see.
On how they voted.
Okay, and then in the background, so let's say you construct this Bell statement and you've been I constructed it in the beginning. Let's say I made a real statement that is, and several people vote on it as they agree with it.
Yeah, you'll get more points. 4 people vote on your Bell statement that you constructed, you get 100 extra points.
Okay, so then also I have a question. Technically, the data is saved as a Bell statement, and I'm not that familiar with Bell, and I'm trying to learn a little bit. So in the background then, is there also the way that saves the the author, the person who entered that Bell statement?
Yes, yes. There are people, the background administrator of the site is able to extract everybody's entry, and that's what we're actually doing right now. We had some preliminary results already, and the team at Solventa and PMI right now is reviewing the entries by different participants that we are planning on inviting to the Jamboree to make sure that the things that they entered are not random.
Okay. Okay. But when you make let's say after the challenge is over and you make all the data available, will a Bell statement be associated with the person who entered it and with the people who voted on it?
I don't think so.
And
once it's finalized at the Jamboree and published and put in public domain, I don't think you will have people associated with who entered things, because a lot of things are entered by Solventa and PMI scientists when these models were first constructed, and then in addition to that, the scientists who participated added new edges and new evidence. So I don't think we're going to keep the authorship of who added what, but we will have the information about the supporting statements, who added what PubMed ID that was added that supports this edge.
Okay, okay. All right. So, okay, that's interesting. That's good to know. And like Bell statements that are already, since you're from Solventa, I suppose you know, Bell statements that are already in the public domain aren't associated with a specific author unless they could be associated with a specific paper, but not necessarily someone who constructed the Bell statement.
No, I don't think so. I think
Okay.
in public domain there are few, one or two small knowledge bases that comprised of these Bell statements. They are there for people to install on their computer and start using them. But I don't think there is a name of a person who constructed the Bell statement. In Solventa, the internal database does keep the name of the person who entered the Bell statement, so we can go back and, because there is somebody reviewing all this. There is someone who enters, and there are people who are reviewing it, and we need to know who to go back to if there is an issue.
I see. Oh, that's interesting. So you internally keep all that data, but that wouldn't be part of the data in the public domain. Okay.
No, I don't think so.
Okay.
Even internally, we don't really keep full name and address. It's login ID, but we know who is who by login ID.
Right, right. And when you get onto this SBV Improver site, is there some sort of verification for how you're known as a real person, or how do you keep control of that it's people, or real people? Is it like they don't enter Mickey Mouse? Is the
we communicate with the people.
Okay.
Actually, working on this project, I know at least half of these people personally now. They send emails asking questions about Bell, asking questions about boundary conditions, send some papers asking, is it an appropriate paper to add to this particular model? And we keep in contact with them all the time.
Yeah, okay.
So if you don't think it's an actual person, then we go and check to see what they're entering. But we haven't seen any non-real people or people who would vote randomly. You can pick that up.
Yeah, but I was wondering, I know at least, I think Jack might be able to comment on this as well, but some people who do annotations for ChemSpider, they use a pen name, or they use a name that's not necessarily their real name, but they do a lot of very good annotations. I was wondering if you had the same thing, and you see that also like on something like Wikipedia, where people who are, their real name isn't necessarily shown.
Yeah, we have some people who registered with just first name. I don't have them as the top 10 users, but we do have some people who registered with the first name only, and that's how they are known. But we
Okay.
still have the email addresses when they include the email addresses when they register, because at the end of the challenge, when we select our top contributors, we need to communicate with them and invite them to the Jamboree.
Okay, okay.
If you don't know who they are, how are we going to invite them?
Yes, I was thinking, though, that if you had massive use of this system at some point by many, many people, you could have, you know, people you don't necessarily really know as their real name, but some sort of pen name, and who could still be contributing good information. So I was just wondering. Yeah.
We haven't had to deal with that.
Yeah.
I wish we had so many people that we didn't know all of them personally, but right now we have so few people. We know most of the top contributors personally by now.
Okay, I see, I see. Okay, well, it looks like a very nice system. There hasn't, have you had any people who, well, had trouble picking up the method of doing the annotation? It looks pretty straightforward. It looks pretty easy, but I don't know if
yeah, but some people put in the wrong Bell statement. That's why we added that drop your text kind of feature halfway through the project. So people, if they are not sure how to construct a Bell statement, they can just send the quote to someone who knows how to do it. But when people construct inappropriate statements, we just email them, flag the statement, and tell them to re-edit in a proper format.
Okay. Is it also possible, so it looked like the Bell statements, again, I'm sorry, I'm not that familiar with Bell, but it looked like the Bell statements were just a triple, so subject, predicate, object.
Yes.
Is it possible to have a Bell statement that, well, let's say, is something a little bit more complicated, like this, for instance, this post-translational modification occurs in this protein at this position?
Yes. Yeah,
Okay.
there are, you can do the Bell statement, and I can try to find an example real fast to see if you can. Yeah, you can never find what you're looking for when you really, really need it. Okay. Here's an example. I open it on my computer. It said that NF-kappa BIA protein modification phosphorylation 332 directly increases degradation of NF-kappa BIA. Basically what it means is that when NF-kappa BIA is phosphorylated on the serine 32, it then degrades.
Okay.
So you can construct a statement like that.
I see, I see. Now, there, you know, in RDF or we in RDF, they usually use URIs for a lot of your things, like here, the Human Genome Nomenclature Committee, NF-kappa B. Instead, we would use, well, if you were writing it maybe in RDF, you might use a URI. Do you have converters already that would convert from Bell to RDF?
I'm not sure about that, but I spoke to a scientist like a couple of days ago at Luxembourg University, and they have the maps. They have a Parkinson's map that they code it in a different language. Right now they want to convert it into Bell, so they write in their own converter, converting their statements into Bell statements. So
Okay.
I'm not sure if there are any converters that are publicly available right now, but I know people are writing them as they need them.
Okay.
The Bell is well documented. There is an Open Bell website, and there is an Open Bell wiki, and it's now being developed by the Linux Foundation. So it's kind of, it's in development. People are contributing, new functions are being created.
And so I'm sorry, I sort of missed a little bit at the beginning, and this was all the data that you used in the beginning to construct your networks and the different challenges all came from Solventa data.
Well, let me get to the slide, how we constructed it.
Yeah.
So this was constructed using two data sources. First of all, we used published data. So we started by selecting a corpus of the literature that would be appropriate for this particular model. We were looking to reflect the lung biology, normal and COPD, so we are not including anything that was studied in asthma, for example, because it's very different signaling. And once you selected this corpus of literature and build a model, then we use that model to add additional edges and nodes from the knowledge base that Solventa has. It's a huge knowledge base, and some of those were not really appropriate because knowledge base doesn't really allow you to filter for like COPD only. So we've added them, then we curated it and deleted some of them. In addition, we also added some of the data-driven nodes that we derived from transcriptional profiling data. This transcriptional profiling data are publicly available, and they represent modulation of lung, pulmonary relevant cells and tissues with environmental inflammatory relevant stimuli. And then we use this transcriptional profiling data and apply reverse causal reasoning that Solventa has, the proprietary algorithm, to derive upstream controllers. Those upstream controllers, those nodes, were also added to these models and were connected through edges derived from knowledge base. And then we did another round of manual curation. And now it's still yet another round of crowd-based curation of these models. Hopefully at the end of it all we will have a good product.
Okay, so basically I think what you're telling me is that it's all human annotated, and then you use you started with human annotated statements and then built a model using those statements. So it's really very strong, let's say. It's not text mining data, it's not that kind, it's all comes from
perfectly curated because we have 50 models here.
Okay.
And one of the steps what we have allowed edges from the huge Solventa knowledge base, and some of those edges were supported by literature we are not really considering within the boundaries. And that carried over. So we curated some of it, but some of them that is being curated right now. Some of the edges that we have included through this automated process of querying the huge knowledge base, some of those edges will be deleted because people vote reject on them because they don't belong in this pulmonary set of the models.
Oh, okay, okay. I got it. Okay, well, it's very interesting, and it looks like a very nice system. I appreciate that you've taken the time to explain it, because it looks very nice. I will try to go and see if I can do some annotation there.
Thank you.
When does the challenge end? I mean, if the challenge ends, are you taking the system away, or is it remaining available so people can continue even if
the challenge will end at the end of February, but the plan is to keep it available after that as well, so people can annotate some more. But for a week or so, it might be frozen because we will need to download everything that will be done by that point, so we can discuss it at the Jamboree in March. So it will until February 23rd. It will be open. Then it will probably be frozen for a few days, but then they will reopen it again for people to continue annotating. And after the Jamboree in March, it may still be open for annotation. Eventually, it will be frozen and put in a public domain as is. But I don't know exactly when it's going to happen.
Okay, but without the annotation component, it'll be, the data will be frozen, but will the annotation component be?
I'm not sure if there will be an opportunity to continue annotating and how it's going to be handled, because in order for people to continue annotating, there should be someone on the other end kind of curating the annotation.
Mm-hmm.
So I think it's still being discussed what would be the best way to keep this still available for everyone to annotate and not have half of the people at Solventa tie up with maintenance of this public resource.
I see. Okay. Okay.
So right now PMI is funding this, so Solventa scientists can spend their time curating and communicating as contributors. But once the challenge is completed, we'll have to find another way of keeping this more or less clean, so you don't get
I understand,
inappropriate annotations. So if you're going to register, I need to make sure that I've showed how to register. So on this Improver website, in order to register, you have to go to the main Improver website, and then here you will have a registration. And then you register, you'll get your email, you use the link in your email to verify it, and then you will return back to the network verification website. There are two websites and people get confused. One website is for the whole Improver. This verification website had to use a different technology, so they built a separate one just for this network verification.
Oh, okay.
So it's
the verification is...
You can access the network verification from the main website where you're registering, but you have to make sure that you get here once you registered.
I see. Okay. Okay, I think I got it.
All right.
So thank you very much.
Thank you. So any other questions?
So I think that's probably it for the questions. So just wanted to thank Natalia Bukharov for a very interesting webinar. And as I say, we'll be putting up the recording of the webinar and the slides on the openphacts